Hi, I'm Mr. Williamson, and this is the first video in the genetics unit. In this video, we're going to be taking a look at the history of the genetics, two of the main principles in the genetics, the rules that we go by, and a couple terms. Before we get started, make sure you're taking good notes. Again, I can't say this enough. I personally think students succeed best with Cornell style. I would use outline myself. That's just me. But you use whatever builds your burger. I'm okay with it. Let's get started. First of all, what is genetics? It's the study of heredity. Okay, plain and simple. Genetics, the study of heredity. How do you inherit traits and things? All right. So as I start every unit, what is the history? How did we get to where we are today? Well, we have been studying a breeding of things for a long time. 10,000 years ago, we have records in the hieroglyphics that indicate that Egyptians bred wheat. In other words, they crossed different strains of wheat to get the best yield, to get the bigger kernels, or to get it to stand up better, or whatever. I don't know, but we have records of that. 4,000 years ago, we know that Asian horse breeders were keeping records of what animals they bred together, or which animals mated, and the results. We've been breeding organisms since the beginning of history. All right, we breed cattle. You do not get this, which is a Texas Longhorn, and a Holstein, which is the cow that gives you milk, without some breeding. We breed corn, crops. We breed corn. We breed other things, just corn. We even breed pigeons. There are people that breed pigeons. They're looking at the heredity of pigeons. How do I know that? Well, one, there's pictures of it, and this pigeon is not going to fly. This is not a natural pigeon. We breed dogs. Look at the different kinds of dogs in the world. How do I know? Because this dog is not natural. This is an hors d'oeuvre in the wild. You let this dog out, and a coyote, wolf, whatever, probably a praying mantis, would eat it as an hors d'oeuvre and still be hungry. This is not a natural dog. We bred it. Genetics. So how do we figure this whole thing out? Well, there's an Augustinian monk. His name was Gregor Mendel. He lived, as you can see, 1822 to 1884. So you got to think about the timeline there. 1884 was about 20 years after the Civil War. So he lived about in the Civil War times, okay? He lived in Austria, which we now call Czechoslovakia, all right? But it's kind of northeast of Italy, and between Hungary and Italy, but there it is, okay? He was an Augustinian monk, like I said, who was in charge of the abbey where he lived. He was in charge of the garden and the orchard. He was the one that raised the food for all the other monks and for the abbey. All right, and I'm sure that he probably had a gated garden because he didn't want people to come in and steal his crops and things. And if he had a gated garden, he probably locked it. And every morning he would get up and he would go to his garden and he would take his keys. Let's do some role playing here, okay? I'm right now, pretend like I am Gregor Mendel. I'm a monk who lives in Austria, and I'm in charge of the garden, and I reach into my pocket and I pull out these. What are these? No, they're monk keys. I'm a monk. These are my keys. They're monk keys. I wait all year to tell that joke also, but you're not there in person, so you get to suffer through it on a video. Anyways, going back to Gregor Mendel here. Monk keys. Name 3 keys that don't open any locks. Get back to that in a moment. Anyways, he did a whole bunch of breeding experiments in the garden, and he published his work in the scientific journals of the time, which was right after, in 1866, and you don't need to know the dates or anything like that, was right after Darwin published his, and nobody cared. All right, but what he published was really important to Darwin's evolution because it explained how it could happen. All right. The three keys that don't open any locks: monkeys, donkeys, and turkeys. Yep. You got to suffer through it even while watching the video. So he experimented with pea plants, just as ordinary pea. I'm going to show you pictures here in a minute. And he chose the pea plant because he could get really, really pure strains. He could get, there were no mixing. It's a very simple plant. It has not a lot of chromosomes, not a lot of genes. And he planted, and look at the bottom down here. He planted and kept records of over 30,000 plants. And when I show you what he did, you're going to think, yeah, well, I can't do that. I wouldn't do it. I wouldn't either. Okay, so what did he do? Well, he planted yellow versus green peas, all right? He crossed them. So in other words, he took pure yellow and pure green, and he crossed them. When I talk about pure yellow, I bet you didn't know this, but peas come in the color yellow also. They come in green. So he took yellow and green peas, and you can see here, all right, and he crossed them. And the offspring, the F1 generation, F1 stands for filial. The filial 1 generation came out all yellow. All right? But then he crossed them. This would be like siblings reproducing. Don't go there. Just trust me. So the parents, the P1, this is the P1 generation. The P1 generation was pure yellow and pure green. And when he crossed those, he got all yellow. There were no green plants at all, no green peed plants, no green peas. But when he crossed those, the siblings, he ended up with 6,022 yellow and 2,001 green. That's the F2 generation. F2 stands for filial 2, okay, which means it's the second generation. It's like the grandchildren, okay? He crossed round versus wrinkled peas. First time when he crossed them, the offspring, all of them were round. None of them were wrinkled. But then when he crossed the siblings, the second generation, the F2 generation, the filial 2 generation, the grandchildren, he got 5,474 round ones and 1,850 wrinkled. What are we talking about when we talk about round versus wrinkled? We're talking about the pea. Normally, we buy peas everything because the American culture likes a round, smooth pea because that looks good. But they also come in this they come wrinkled. I don't know if you knew that. All right. He looked at whether or not the pods were inflated or constricted. And over here you can see these are constricted. You can see the individual peas in them, whereas these are inflated. The peas you can't see, all right? So he looked at those. And you can see where when he crossed them, he got all inflated. But when he crossed the offspring, the F2 generation, when he crossed the F1 generation, two F1s, put them together, he got the F2 generation, the grandchildren, 882 inflated, 299 constricted. He even crossed yellow versus green pods, not the color of the pea, but the pod. All right. And he looked at, and that would be down here. When he crossed those, he got all green. So if you're really paying attention here, the peas, when he crossed them, he got all yellow. But when he crossed the green pods with yellow pods, he got all green pods. All right. And he ended up with 428 green and 224 yellow. He crossed all kinds of things. Here's a little table including the color of the color of the flower, purple and white. He colored where the flower location is. Axial means it comes out from the middle of the branch. Terminal means it comes out the flower is only found at the end of the branch. And he talked about how plant size, tall versus short. All right. And he got this table. Again, looking at the table, you can see the first generation. He crossed every one of these were pure. The P1 generation here, the parent generation. There is no P2. Don't ask me why they call it P1. There is no P2. But the P1 generation were pure. So it's pure long stem, pure short stem, pure axial, pure terminal. There was no crossing. But when he crossed those together, the pure and the pure, he ended up with like all long or all axial or all round. But then when he crossed those together, it went back to this here. Now I'm going to ask you to pause the video here just for a moment and see if you notice a pattern in this second column, in the F2 generation. Do you notice any pattern? Do you notice any pattern between the F1 and the F2 generation? Like the numbers here. And well, there's no numbers here, but the pattern from what you can see here and the numbers on this side. Do you notice a pattern? All right. Then I'm going to ask you about the ratio. The ratio is that final number. All right. So we're looking here. What is the ratio of yellow to green? And ratio, find the ratio is real simple. We're going to be doing this in class. Take the smallest number and divide it by both. Well, 2,000 divided by 2,000 is 1. 6,000 divided by 2,000 is 3. So it's like 3 to 1. And that's what he figured out. Almost all of them are all 3 to 1. If we took the ratio of everything over here, they're all 3 to 1. And if you notice, whatever's over here is the larger number over here. The round, bigger number. Inflated, bigger number. Long, bigger number. Colored, bigger number. Axial, bigger number. Whatever was shown up as the F1 generation was the one that had the most in the F2 generation. So, why? Why did all that occur? All right? Because the parents each had a gene for the factor, or he called them factors. We call them genes now. We're going to talk about that. Let's say green and yellow. That's what I'm going to be using the most. One gene, one of those factors, is dominant over the other one, over the recessive gene. It's the one that shows up. So you have pure yellow. So these are two. We know we get two factors, two genes, two factors, one from mom, one from dad. We talked about that last unit. And green here. So we know that mom is going to, remember meiosis, you pass on one of the pairs of the chromosomes. So let's just say, pass on that one, we pass on this one. Doesn't matter, they're both the same. Everyone in this generation here is going to be YG. One from mom, one from dad. They're not YY. You have to have two. You have to have two because one from mom, one from dad. All right? But the second generation, F1 generation, is all YG. And because it's YG and because Y is dominant, yellow is dominant over green, we don't see any green. But when we breed these, we get this 3 to 1 ratio. 3 yellows to 1 green. We're going to have to figure out why that is. All right? That brings us to Mendel's first principle, the principle of dominant and recessiveness. Write this down. Each trait is controlled by a pair of factors. This is his words. We call them, shouldn't call them chromosomes, we call them genes, actually. Each factor, each trait is controlled by two factors, one from mom, one from dad. The dominant factor covers up or hides the recessive factor. A lot of people think dominant is better or stronger because in some terms in our language we use the word dominant as meaning that. That's not what this means. It just means it shows up. Brown eyes are dominant to blue eyes. Doesn't mean they're better, doesn't mean they're stronger, just means that when you have a blue eye gene and a brown eye gene, the brown eyes show up. Yellow peas are dominant to green peas. Doesn't make them better, doesn't make them worse, doesn't make them stronger, just means that yellow, when you cross a yellow and a green, you're going to get the yellow one show up and the green one won't. Also, we look at Mendel's second principle, the principle of segregation. Heredity factors are determined by here's the part that you need to forget reading all that stuff, okay? The two factors segregate. This is meiosis. This is why I put meiosis before this unit, because we had a Y and a G, and we're on each chromosome. So each chromosome here, they replicate, blah, 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 prophase, metaphase, anaphase, telophase, all right? And now we have them into two, and then they separate again, and we end up with shouldn't really be X's, but that's okay. We end up with one of them, All right? You get one from mom, one from dad. Mendel's second principle says that you're going to separate those when you make gametes, when you make sperm or egg, when you make sex cells. We're going to separate it. You have a pair of chromosomes, one from mom, one from dad. When we, through meiosis, you should know this, all right, that they separate. We're going to pass on one of those genes, either mom's or dad's, or maybe, because of crossing over, a combination. But for each trait, each thing we're looking at, yellow colored peas or green colored peas, we're going to pass on one of them. We're not going to pass on both. Today we call those genes, okay? And each gene is located on a different chromosome in the homologous pair. So when you look at a karyotype, remember the karyotype, the table of chromosomes, the big ones, all the way down to the small ones, with the X and X or X and Y, all right? Each one of those pairs contains, on each chromosome in the pair contains the gene for that trait. We're going to separate those. Now we call those alleles. Instead of traits, we might call them alleles. All right? Not alleles. They're alleles. They're traits. Traits or alleles. Either word, that would work. Traits. For instance, by the way, trait could be eye color, hair color, how tall you are. It could also be how well do you digest some food. Like, for instance, are you lactose intolerant? That's a trait. You can have a trait, how much do you sweat? How whatever it is. Everything in your body is controlled by genes, by heredity. Okay? Which brings us to a whole another little couple of language thing. I'm going to be using these two words a lot, so you need to know them. You need to know them. Genotype versus phenotype. The genotype, we're looking at the genetic makeup. So for instance, in the peas, we might be looking at YY. That's the genotype. Or maybe we're looking at YG, which would be yellow also. Or we could be looking at GG. Those are the genotypes. You cannot see the genotype. I cannot look at you and determine your genotype. Okay? Like if you have brown eyes. I don't know if you're brown, like pure brown, or I don't know if you have like parents, one who has blue eyes and one has brown eyes, or I don't know. I can't see your genotype. All right? But I can see your phenotype. Your phenotype is what you look like. Your eye color, the color of the pea, whatever. I can see the phenotypes of these kittens. All right? We have a really dark kitten here, and we have a light kitten here. That's the phenotype. Now, I don't know the genotype. I don't know what's dominant. Let's just pretend for a moment that dark is dominant. Okay? I don't know if this is, because this could be, if we're looking genetically, this one right here could be dark, so it would be a dark cat. It could be a dark light. You got a light gene from mom or dad, but from the other parent got the dark gene, and because dark is dominant, the light never shows up. I do know this one is probably light, because, again, if and I'm making this up, if dark is dominant, the only way to have a light-colored kitten would be to have two recessive genes. So, but I cannot tell the phenotype. The phenotype I can see. It's a photograph, and that's what I've had students tell me, that a photograph, the word photograph, starts with PH. Phenotype. Starts with PH. You can see the phenotype. You can see the photograph. The genotype is the genes. So let's go back to Gregor Mendel's second principle. All right, and first, technically. If we have the cross here, we know, like I said before, that this is YY and this is GG. All right, we all know that this is YG, this is YG, this is YG, and this is YG. How do I know that? Because they had to get a Y from one parent. That's the only thing the parent could give, this one. And they had to get G from the other parent. It's the only thing the parent could give, G. So we know all of those are YG. So the question is, how come this ratio down here is 3-to-1? And I'm going to answer that really quickly, and it's pretty much the last thing in the video. It's called a Punnett square, and we're going to go into detail on how to do this. But I'm going to take the parents. We crossed a YG with a YG. We crossed these two. That's what we were crossing here. So the parents, one's YG, the other one's YG. Well, what can the parents give? They can only give one. You don't pass on both of them. The law of segregation. All right? If this parent, what can they give? They could give a Y or they could give a G. They can't give both. This parent can give a Y or it can give a G. Can't give both. You can only give one. So now we just look at what are the possible combinations that each could have. Well, if this parent if the top parent gave a Y, and the side parent gave a Y, then we would have a YY, and because Y is yellow is dominant, it would be a yellow colored P. If the top parent gave the Y and the side parent gave the G, we would have YG. But because yellow is dominant, it would be yellow. Or if the top parent gave a G, but the side parent gave a Y, it would still be YG or GY. It doesn't matter what order it's in. But because yellow is dominant, it would be yellow. But if both parents gave the green gene, then we would have GG. There is no Y to be dominant. The recessive trait shows up. It would be green. Notice this is 3-to-1. 1, 2, 3. 3 yellows, 1 green. Just like right here. 3 yellows, 1 green. So we can look at this and we can go, okay, we know that one of these is probably YY. One of these is probably YG. The other one is YG. And this one is GG. That's how we get the 3-to-1. Hopefully you understood that. Looking at the Punnett square. That's how we got it. We're going to go over Punnett squares in the future. All right, done with the video. Hopefully, if you have any questions, you're going to ask. Just a real quick review. We talked about the history of genetics and Gregor Mendel, and he was an Augustinian monk. These are monkeys. The sad part is that in real life, in person, I tell that joke, and months later, I can ask about what are the phases of mitosis, and nobody remembers. But I can pull a set of keys out of my pocket and go, what are these? And everyone goes, monkeys, because that's the part you remember. Going on. Gregor Mendel, he worked in the garden. He used the pea plants because they could produce pure strains, YY, GG, etc. He crossed a whole bunch of them, kept really good records, came up with 2 principles. The principle of dominance and recessiveness, where one gene is dominant over the other. It doesn't mean it's better or anything. It just means that's what shows up in the phenotype, the thing that we see. And he talked about the law or a principle of segregation, that the fact that you have these 2 factors, genes, these 2 genes, for every trait has 2 genes that you have, one from mom, one from dad. And when you make your sex cells through meiosis, sperm and egg, you're going to pass on one of those genes, not both, one. That's the law of segregation, the principle of segregation. And finally, we talked about genotype and phenotype. Genotype is the genetic makeup of the organism. YY, YG, GG, if we're talking about the P colors. Phenotype is what does it look like. It's yellow or it's green. Remember, you can see the phenotype like you can see a photograph. The genotype you cannot see. It's the genetic makeup. If you have any questions, please make sure you ask me in Edpuzzle or in the classroom. I hope you took good notes. Good luck.